Hi everyone, welcome to my channel. Have you ever wondered how scientists sort thousands of different viruses into groups? Today we are diving into virus classification based on their nucleic acid or whether they are enveloped or not. These two features, DNA or RNA, and the presence or absence of an envelope are like the virus fingerprint. They determine how the virus infects the cells, spreads, and even how we fight it with vaccines and antiviral drugs. Now let's discuss the classification of the viruses based on the type of nucleic acid they contain, that is RNA viruses versus DNA viruses. Let's start with the DNA viruses. DNA viruses, it can be either double-stranded or single-stranded. In the case of double-stranded DNA viruses, they can be either small circular viruses or large linear viruses. In the case of small circular double-stranded DNA viruses, it includes polyomaviridae, papillomaviridae, and heparinaviridae. Their genome size ranges from around 3 to 8 kilo base pairs. And the large linear double-stranded DNA viruses include herpes viridae, pox viridae, asva viridae, iridoviridae, and adenoviridae. These have much larger genome. For example, in the case of herpes viridae, ranges from 125 to 235 kilo base pairs. Now moving on to the single-stranded DNA viruses. They can be also linear or circular. The linear single-stranded DNA viruses include the parvoviridae family, which are negative sense and have a genome size around 2.8 to 5 kilobase pairs. And the circular single-stranded DNA viruses include two families, circoviridae and aniloviridae. The circoviridae, which has an amphisense genome, and aniloviridae, which are of negative sense. So overall, DNA viruses shows a lot of diversity, not only in the genome structure and size, but also in the sense of their nucleic acid strands. In the next section, we will compare these with the RNA viruses. Now let's move on to the RNA viruses. RNA viruses are classified based upon number of RNA strands, whether it is single-stranded or double-stranded, and their polarity, whether they are positive sense or negative sense. Let's start with the double-stranded RNA viruses. They containing RNA in two complementary strands. Example include Birnaviridae, which has two genome segments, and Rioviridae, which contain 10 to 12 genome segments. Next, we have our positive-sense single-stranded RNA viruses. They are special because their RNA can directly act as messenger RNA, meaning it can be immediately translated into viral proteins once inside the host cells. Example include Picornaviridae, Calci, Astro, Toga, Flaviviridae, Coronaviridae, Artriviridae, and Retroviridae. Among them, the Retroviridae family is very unique because their genome is diploid, meaning they contain two copies of positive sense RNA. Now let's look at the negative sense single-stranded RNA viruses. They cannot be directly translated. Instead, they first need to be transcribed into an mRNA by an RNA-dependent RNA polymerase that usually coded by the virus before protein synthesis can occur. That's why it is called negative sense single-stranded RNA. An example for the single-stranded RNA viruses include Rhabdoviridae, Paramyxoviridae, Filoviridae, Bonaviridae. An example of segmented single-stranded RNA virus include Orthomyxo, which is having its segments ranges from around 6 to 8 segments, and the Bunyaviridae is having 3 segments, and Arenaviridae with 2 segments. Overall, the genome size of RNA viruses can vary widely from very small of around 2 kilobase pairs in the case of Delta virus to more than 30 kilobases in case of coronavirus. So RNA viruses show incredible diversity, not just in structure but also in how they replicate and express their genes. Now let's look at the classification of the virus based on whether they have an envelope or not. First, the non-enveloped viruses. These viruses are made up of only viral protein and nucleic acid, either DNA or RNA. The viral protein that make the capsid layer of the virus, and the DNA or the RNA, it is a genetic material. So in this picture you can see the outer, this is a viral protein made capsid, and inside is the genetic material. But definitely they lack an outer covering or the lipid envelope. So it is non-enveloped viruses. For example, among the DNA viruses, the non-enveloped one include circoviruses, adenoviruses, papilloma, polyoma, parvoviruses. Among the RNA viruses, we have the calci, astropicorna, birna and reovirus. Next, the enveloped viruses. These viruses have an additional outer covering, the lipid envelope, which is derived from the host cell membrane and containing viral glycoprotein. That's the thing we have to remember in the case of enveloped virus. Its outer, this lipid envelope is derived from the host cell membrane, but it having viral glycoprotein incorporated over it during the replication. So these structures include viral protein that form the capsid, nucleic acid can be either DNA or RNA, and a host-derived lipid envelope. Examples of the enveloped DNA viruses include herpes, asfar, iridopox. All the bigger viruses in the DNA group are enveloped. And the RNA viruses such as orthomyxo, rhabdope, toga, flavi and paramyxo. Sometimes minor host proteins or DNA may also be present, but they are not essential for the virus. Finally, let's see the significance of the envelope. So these enveloped viruses are very sensitive to organic solvents like either chloroform, alcohol and detergent. They can disrupt the lipid layer and inactivate the virus. So in the case, while choosing disinfectant, we have to look into whether the virus is an enveloped or non-enveloped. In the case of enveloped viruses, it can be easily inactivated by the organic solvents, and in the case of non-enveloped viruses are much more resistant to these solvents, which make them more stable in the environment. So in short, the presence or absence of an envelope not only defines the viral structure but also affect the virus stability and resistance to disinfectant.